It's Can you hear me? Yeah. Okay. We actually have a 4-hour session, and it's really every minute we scheduled. So I know more people will come, but I would like to start. So my name is Jeff Xia, and we have developed MetaboAnalyst. So it's been very popular, and we keep adding a lot of functions. So today, the workshop, we are going to focus on the latest progress. Is for LCMS spectral processing, statistical analysis, and functional interpretation. So this is, we try to get the most recent, up-to-date, and most commonly used, also the section that receive most questions to condense into 4 hours. Usually taking 2 days, so we understand it's hard, but we try to be as smooth as possible. And these slides, we try to acknowledge all the past 10 years, more than 10 years of support from Genome Canada, NIH, and NZERC, and Canada researchers, and more recently from TIMIC. And here's 3 TAs, and all sitting here, Jiang, Jessica, and Yao. And they are my postdoc and a PhD student. They are the actual person developing the tools. So if you're really asking code specific details, and I will try my best, but they are really the person writing the lines of code, okay? So here's the schedule. And you can see it's full. The first 15 minutes, including, excuse me, now it's about opening lecture about the general concept in data analysis and the design overview of MetaboAnalyst. Then the next one going to start with LCMS spectral processing. This is kind of we introduced in the version 5, and that allow people to really just drop your data there, and it will process using optimized parameters. So it's used heavily. We also receive a lot of questions. So it is about 20 minutes. Then you are going to use our example data to do the hands-on practice, because raw data processing actually is, taking time. And if we're doing this, just try to use our example data so everybody has experience rather than download, upload, it taking more time. So we can from beginning to end. After that is statistical analysis. Statistical analysis is very heavily used. So this section is actually the old statistical things, but it's we just commenting on a key concept, because if you're new to MetaboAnalyst, you definitely need to use this statistical module. So we're going to emphasize what's the tips, procedures. And this is section followed by about 20 minutes hands-on. Then have a coffee break, about 15 minutes. I just confirmed they are going to do coffee offered, and probably no snack, but coffee will be there. And then follow that is functional analysis. So functional analysis is how do you get from compounds or peaks, actually get pathways and functions. And this is, we receive a lot of questions, and here we are going to introduce what's the concept, what's the parameters, and then followed by your practice, okay? And the last part is the latest addition for complex metadata, because metabolomics is now used for now is for a lot of clinical or population study, and you have a lot of the experimental factors in addition to the main like control disease. So when we're doing data analysis, we need to account for this covariate. So how do you do this? How do you upload the data and tell the program, here's my experimental design. What's the best way to analyze them? And so this is the last session. Then finally, I'm going to conclude and summarize what's the questions, what's the learning, what's the future, okay? And the next few slides, I'm going to mention about the general concept behind the omics data analysis. And this is very true. Our group not only developed metabolomics, we actually working a lot on microbiome and RNA-seq and even multi-omics. And there's a strong pattern associated with all the omics. So if you understand this part, you can easily understand the other, like RNA-seq microbiome data analysis developed using tools developed by our group. And so all the omics data generation is from the high throughput, high dimensional, like microarray sequencing, mass spec. Then you get a data which you can think about big Excel spreadsheet that 10,000 rows and hundreds of samples. Then you need to apply stats visualization to understand the insight and knowledge. So this is really the tip is where MetaboAnalyst or our tools try to help. And there's new challenges, multi-omics. So if you have multiple tables, how to do that? This is our latest effort. We are going to touch a little bit in my summary in the late. But today we are going to focus on metabolomics. So metabolomics data analysis is the same thing with other omics. We have two major challenges. One is raw data processing. Raw data processing, if yesterday you took the workshop like using MZMine and other popular tools, and it's challenging, but it's getting better and standardized. So you upload your generate your open format. You use this MZMine or other tools to process your spectra. It's taking time, but more or less it's computing taking time to processing data by data, and it should be automated. I'm just always envision like driving using Tesla. So more or less it's going to be standardized. This is our concept. Raw data processing takes time, but it should be standardized, automated, and shouldn't be a challenge. Most challenge is the second one, the complexity challenge. When you get a table and you need to understand data, and it's very hard to automate because we don't know your goal, we don't know your context, we don't know how the other related information comes into play. So this part must be open-ended and it be user-driven. It's by you. And this part is require a lot of time input. It cannot be automated, but it can be made much smooth. So this part is actually most of part we talk about stats, visualization, functional analysis. But the first part is critical. If we generate garbage or sub-optimized data, you are going to all the downstream are going to be affected. So the raw data processing is critical, but it should be standardized. That's my emphasis. So a lot of you just keep in mind the raw data processing. The final goal is generate that table, okay, high quality table, and so our tools developed in the past to address this aim is actually just really fitting for this. As raw data processing, we use a big cluster and in the local because it's really saving some time, and it takes like minutes, not minutes, 20 minutes or even several hours, sometimes longer. We generate a table. Once you generate a table, you can upload to the server, usually host on the cloud, and now you can interact with your data through the web browser. Finally, it's you to making decisions, to writing the papers. So this is what we have designed in the overall, how the tool is supposed to and overlay with the raw data and the stats and with the technology. So everything benefit. We use the cluster, we use the cloud, and we use your browser, and finally it's your brain. So everything have to be working actively to solve that problem. And how do we solve the data analysis things? So I mentioned raw data processing. This is really try to generate a table, and there's two major approaches. There's MS data processing. You learned yesterday, and MZMine, and there's OpenMS. There's MetaboAnalyst have the raw data processing, and the other one is the next generation sequencing. So more or less, you can think about two major things: sequence and spectra, right? So they are getting better and standardized. And next one is after you get a table, what you're going to do? You need to do a quality check-in, batch effect adjustment, and missing value imputation. We are going to touch it today. And after data is cleaned and normalized and fit, and you're going to stats on it, machine learning on it. So this part is very diverse, and it's almost infinity. And so you always need to learn what's the advantage, what's the limitations of different approaches. So from here, you need to find the patterns, and from these patterns are significant features, and you want to get functions or insights and hypotheses. So keep in mind that omics data analysis is always open-ended, and there's no fixed path. So always is driven by your own understanding. It's more exploratory. So independent target analysis follow this. So if you use this very stringent, how to say, the requirement, asking omics data, sometimes it's just a misfit. Okay. So here is the common steps in omics flow. I mentioned about the 4 major steps, but each step actually contains multiple smaller focus. And raw data processing, we have quality checking and missing value, filtering, normalization. This is one we are going to touch in section 2 for the simple statistical analysis. And then we have this univariate, multivariate analysis, and everybody understand PCA, PLSDA. We are going to re-emphasize how to use it, what's the potential challenges. And we are not going to touch too much on machine learning and AI and deep learning. This is not a focus. What we try to do is introductory to prepare your data, which is ready to be interpreted. Even you doing this fancy machine learning, you have to start from basic understanding of the data. What's the quality, distribution, do some t-test ANOVA, and do some simple thing to understand it thoroughly before you're doing more advanced. Okay, this is very, very critical. Using simple, effective, robust method to understand your data first before you apply any advanced one. So one I usually don't like is people don't have too much biological, medical background, just purely computer science, machine learning, and apply every fancy tools on your data. And then talking about exciting discoveries, which is very dangerous. So we need to understand the data from simple, effective method first before we try other method. So data interpretation really truly subjective, but subjective means you use your background knowledge. When you're writing a paper, you need to talk about the context, talk about the literature, all the things combined together to writing that beautiful paper. Do not rely on p-value alone. So this is something I always want to show in my presentation is you are at the center, you are the master, you are the driver. We develop tools, it's for you to help you, and we don't give you a final answer, it's you to decide. And also stats also is not the final say, stats or p-value not significant, my result is not useful. No, it's not about that. It's really about your understanding of the data and your design, and how it compared with the literature. So your brain needs to work actively. That's the one. It's enjoyment of the research. So so now let's go to the MetaboAnalyst. And MetaboAnalyst been there for more than 10 years. At the beginning, we focused on the stats, and clearly at the moment, there's no other statistical tools. And the motivation is that, hey, we want to do something, make it useful online. And with the years, it became more popular. And then we started doing more like, what's next? So we get the signal features. We want to do more functional analysis. But at the time, what we try to do is mainly about target approach. We have to identify the compounds first. And in order to do data analysis, we have pathway analysis, enrichment analysis. And then since version 3, we became more focused on translation and raw data processing. So biomarker analysis in version 3, and later 2018 and to version 4 and 5, we gradually move into untargeted analysis using LCMS for functions and raw data processing. Why is because all the previous questions we try to address have been solved. Okay, I'm just saying solved. It means in method-wise, you can continue to improve, but the major bottleneck is solved. But the raw data processing, how to do the on-target analysis is still a challenge, so we continue doing it. Actually, we are going to release version 6 soon, and we are doing MS2. We are going to talk slightly more details later. So this is continuing the issue. So it's being used widely, and hopefully going to be used more in the future. And here is the overview of today's modules. We're going to talk on 4, is raw data processing, and section 2 is simple statistical analysis about data processing normalization. Then section 3 is functional analysis. How do we interpret in the peaks and to functions? The last one is complex stats. Basically, how to do a covariant multi-factor analysis. And we do have these protocols. 4 hours is not enough, clearly, but we do have very detailed protocols. One we published last year for Nature protocol, which is really focused on 2 sections today, raw data processing and the multivariate analysis. For stats, and we do have another protocol published for version 4.0, which is still relevant, okay? And so more or less, you can follow them to do your analysis. So even you missing something, you download this paper, you just follow, okay? Logistics, I already print out this one and put it in front of everybody's table. And you should be able to log in. And do not use the free Wi-Fi, which is too slow. You have to use this one, otherwise everything's slow. And we put all the materials on the GitHub, and so it's like ShareLab 2023. So we have several servers set up. You don't need to care, just go to the main server, it will automatically redirect to our new servers. And the last one is forum. And I understand we won't have time to cover all the questions. Just register your name and post your question there. So either my team or other people will help you. So I realize it's really overwhelming, too many for my team to handle every question. So everybody help each other. Also, our answer can be benefit other people. So search the forum before you ask. And caution and recommendations. This is, I don't know it's going to happen today. Sometime if too many people use it simultaneously, and another workshop, potentially in Europe and China, they are also having at the same time. We have like several hundred people using the server simultaneous, could be slow. I hope it's not the case today, but it do happens sometimes 400 or 500 people. Suddenly we don't see that happen several times. And to reduce that, if you also hope you can make new friends, is form a group, two of you. One is display the either the slides or the tutorials. The other one is just go click the buttons. So it's usually best. One screen for the slides, one screen for the just MetaboAnalyst. Okay. This is forum, and it's free, so you just register and ask questions. And the next one is a raw spectral processing by Chiang. He is my PhD student. He developed the whole workflow for the raw data processing. I think he will give you the lecture followed by your hands-on practice. Okay, thank you.
So thanks everyone. Thanks Jeff for the brief introduction. I hope you can hear me fine. So good morning, everyone. I'm Zhiqiang Pang. Today I'm going to show you LC-MS spectral processing by using MetaboAnalyst 5.0. So I come from Jeff Shah's lab, McGill University, Montreal. So now this is the first session for today's workshop. In this section, we are going to cover the LC-MS spectral processing. I will give some lectures on the basics, from something very basic to the deep understanding on the raw data processing, as well as some demo. Then I will give you around 20 to 25 minutes for hands-on. You can practice according to my demo to see if you can experience some problem. So this is the section 1. So let's get started. So now when we're doing the chemical analysis, we are the first question for us is how are molecules are different from each other. So the very basic things is that they have different mass based on the types of their items. For example, CO2 is so different from CO. So this is where they are different from, and we can identify the molecular based on these rules, basically. So how we measure these things by using the LC-MS. So LC is the liquid chromatography. This is a very common analytical chemistry approach to separate the compound by using mobile phase. As you can see in this, in the column, the compound will be eluted by the column, by the different mobile phase, and different polarities of the compound or different characteristics will be eluted accordingly into the mass spectrometry, which is named as a detector. They can detect the different amorphism of these ions. So this is the method named as LC-MS. This is something very basic. Traditionally, we have a lower resolution mass spectrometry, and they can measure a certain range of the amorphism value. But in recent years, the higher resolution have been developed. We can identify the specific molecules or ions from the LC column. And the higher the machine resolution, the more we can narrow down the possibilities for the different ions into their very narrow amorphism value. For example, here I give an example, 157.318. For this amorphism, we can find out the potential formulas for this amorphism. We have two candidates. Compared to the very wide amorphism range, we can clearly narrow down the possibilities for the chemical compound. So this is the high-resolution mass spectrometry we have been using in recent years to increase accuracy for compound identification and feature detection. So what does the MS data looks like? So MS data including MS1 and MS2. Here I would like to emphasize the MS1 data because in the MetaboAnalyst 5.0 we mainly focus on MS1 data processing. So in the MS data in the MS spectrometry, we will get the compounds eluted from the column. It will be detected by the detector, mass spectrometry machine. The detection happens every just milliseconds. So as a result, there are going to be a lot of scans. They scan the component from the liquid chromatography every millisecond. They will generate a lot of scans in their data. So this is the data. This is the MS1 raw data structure. So based on the MS1 data generation process, we can get a general chromatography profiles. As you can see, this is the total ion chromatography of the MS data. In this data, we can see according to the retention time, the different ions have been eluted from the liquid chromatography. Different intensities will be detected. So this is the sum intensity of all ions. When we go into the details of this data, we can find out more information from another dimensions, amorphism. We can see in the same retention time, there are clearly different amorphism value there. This is called MS feature. We have to find out these MS features because they are highly related with the compounds information in this data. So now let's talk about something about the raw spectral data processing. The first thing is that we got a data from the mass spectrometry instrument. We have to convert the data from MS instrument into the metabolic features. This is a process called spectral data processing. So for the mass spectrometry data, it usually contains 2 to 3 dimensions. For example, for direct injection MS, the raw data include the amorphism and intensity dimensions. This is the 2D data. And for LC-MS, usually there's another dimension named as retention time, including, so there, this data including M over Z, retention time, and intensity. This is the 3D data. So the most common vendor raw data file formats are .raw, .wif, or something else. This is the generated by the instrument directly. So you can find out find the data from your vendor software, vendor acquisition software directly. But when we got this data, we can use the commercial software to do the processing, which is the black box. We don't know what's going on inside. If we want to use the open source software, which is usually better than the commercial one, because everything is open, it's having contributed by the developers many years. We have to convert the data into the open source format. So convert the data format into open source format is the first step for the raw data processing. In this step, we have to figure out a question about what's a profile data and what's a centroid data. So usually the vendor raw spectral data is usually in profile format, which is redundant for the regular LCMS-based metabolomics analysis. We need to convert this kind of profile MS data into centroid mode to condense the Gaussian profile peaks into the centroids. As I have illustrated before, there are going to be a lot of scans when we're doing the data acquisition. So the MS spectral instrument will detect every single details from the columns, and they will generate a Gaussian peak inside a single scan. We have to find out the most critical part for this, for specific M over Z. So this process is a centroid. We condense the Gaussian shape into just one point. After the centroid, the data complexity will be reduced significantly, and the data processing will be accelerated without almost nothing lost. So the most common open source format for raw spectral data is the .m over z ml and .mz xml. This is the two most common format, and there is also something else, for example, .cdf, .mz data, or something. So for the open source software for raw data processing, there are a lot of very commonly used softwares. XMS Online, XMS Bioconductor package, MSDAO, MZMine 3.0 version. This is the latest version for them. And also OpenMS. This OpenMS is firstly developed for proteomics. Now it's also being used for metabolomics. So this software, they are using different algorithms to do the raw data processing, and the result is also different. For the algorithm they are using, the most commonly used one is the SandWave. This one has been developed many years ago, have been also updated in the past decades. This algorithm focuses on the peaks, the regions of interest in the MS data, and they will find out the peaks that have a very nice peak shape. Usually, the peak shape can have different peak wise, but to get the best result, there are a lot of parameters for this algorithm to run. As you can see in this figure, at least 8 or 6 parameters need to be optimized to get the optimal parameter combination for your data. So the most critical question here is that, can we make this process automatic? So to resolve this issue, we develop an auto optimization workflow in MetaboAnalyst. So our optimization approach is designed to extract a region from the data, from your data. You upload the data, we extract a specific region abundant with MS signals for design experiment-based optimization. So in this step, we extract the most abundant part from the data, and we ignore the sparse area only for the parameter optimization. So let me go into a little bit details on the auto-optimization workflow. So firstly, we will get the data, as you can see in the panel A. This is the really raw data, including everything. We use a sliding window inside of the different dimensions, as you can see in B and C. We will select the regions really abundant with the MS signals, and we will extract these regions for our parameter optimization. As you can see in this region in the panel D, both low intensity peaks as well as high intensity peaks will be retained for parameter optimization. Then we use a very complex DOE-based parameter optimization. DOE is a design experiment to optimize the 8 parameters within 44 runs. If we want to optimize the parameters one by one, it's going to be over 6,000. So by using this design, we reduce the optimization step a lot, and by increasing the performance. And then we evaluate the quality of parameter optimization by using the formula. We will consider the peak shapes, peak stability, as well as the isotope informations inside the data. So I would like to share something on the performance for the parameter optimization. Here I use the 3 data sets, including standard mixture, NIST data, and the IBD data, which is the real biological data. Our algorithm is named as OptiLC-MS, the third one. As you can see, compared to other optimization approach, OptiLC-MS could run very fast in just minutes level, and the results also getting much better. As you can see, we keep the total peaks don't increase too much, but we have the highest number of the true peaks and quantified consensus peaks, as well as the highest peak ratio for Gaussian shape. And then we also benchmark our studies by using the biological data. It's a malarial data set from our collaborator. We compare the results in different aspects. So compared to the default parameter and the optimized parameters, as you can see, we can clearly increase almost every aspect for the data processing results. Then I also benchmarked our algorithms by doing a dilution series. I use a mixture of serum and urine in different ratio. As you can see, we can clearly see a dilution, serial dilutions in the heat map compared to other common softwares, XMS, MS-DAO, and MZMine. MetaboAnalyst could find the highest correlation coefficient. So this is just briefly introduced some benchmark studies. So I would like to summarize a little bit. What's data processing, pre-processing of LC-MS data? In one graph, as you can see here, we just try to find out the peaks from the raw data in the different dimensions to make the peaks high quality and reasonable by using auto-optimized workflow. So the ideal result for us, we will got some summarizing peaks. This is a result from MetaboAnalyst. We are going to show you later. In the results, we can have the retention time of the peaks as well as the ambiguity values. So for the different samples, we are going to have the relative intensities here. So this table can be used for different purposes. For example, stats, biomarker analysis, and the functional analysis. My collaborators will introduce a little bit later in the later section. So here is the MetaboAnalyst 5.0. you can use the URLs and go to the home page by clicking Start Here. You will find this module page. The first module listed on the top of this page is the LC-MS spectral processing. This is a module we are going to demo a little bit later. So in this module, we include a lot of functional utils, including the raw data uploading. We allow user to upload the MZML, MZXML, MZ data, and CDF. This is the four most commonly used open source data format. We also support them to do the centroiding on the fly, but this is not encouraged because uploading our profile data is really slow. It's going to be gig level. We don't support that kind of large data uploading. We support at maximum 200 megabytes uploading. So it's better to do the data centroiding at your local to convert the data into centroid mode. And then we can do a parameter optimization automatically, and we will got the optimal parameters. After we got the optimal parameters, we will do the picking, alignment, gap filling. So after everything is done, we will do the pick and annotation to annotate the MS features into adduct and isotope level to see the relationships between the different MS features. And finally, we will do the putative compound mapping from the MS1 level. The result can be visualized from a very nicely designed page. So this is the page we have designed in a little bit early version. So it's have been updated. But here I want to introduce a little bit on the total ions and extractive ion chromatography. So in the final result page, user could click the different, could view their result in this 3D plot. It's a score plot and loading plot by showing their different features. They can double click the nodes directly. It will generate a total ion chromatography of this file. For the different loadings, that means that it's different MS features. They can click and select the features according to their importance and click them and see the real stats and the EIC extracted and chromatography will be plotted interactively. After the raw data processing, it's going to go to the final page. User could download the result directly, or they can use the results generated by the raw data processing module. They just select a different module and click go. The data will be transferred to the different modules. They can do any exploration online directly. So here I'm going to do some quick demo on how to do the raw data centroid and conversion. I will also do raw spectral data uploading and parameter setting and do a quick demo on the result exploration from MetaboAnalyst. So everybody, if you have the laptop, you can find out some raw data from the GitHub we have shared with you.
Are you sure you want them or directly within the?
No, I will show quick demo. It's very quick.
Yeah, you're asking people to follow or not?
No.
Okay.
No. Yeah, I'm not asking you to follow. I just want to show quickly, and you can go later, right? I will work around after my demo. So if you have any questions or need any help, just hands up later, right?
So we're running a bit late, so just doing demo, then we just basically let them know.
Yeah. So the most commonly used software to do the date conversion is the MS Convert. You can download it from the ProteWizard software. This is the very commonly used one. it support almost all raw data conversion. So here I would like to show a little bit quickly how to do the data raw data conversion and centroiding. So here I provide 3 raw data. It's from Thermo Fisher, and we can quickly select all of them. And many people have a lot of questions about the data centroiding, how to do it. I want to, because for MetaboAnalyst, we only support the centroid mode. So this is the most commonly asked question. Here I want to quickly show you, we have to select the peak picking and add this peak picking condition as the first one. And we can do in the data conversion in the parallel mode. So you don't need to follow. You just see what I'm doing. And we click start. It's going to be converted directly. So it should be very fast. After we finish the conversion, we can see the result will be something like this. It's going to be MZML. It's really small. Usually the raw data is less than 100 because this is a very complicated serum data. So, but after the conversion, it's become very small. It's stored online, uploading and processing. This is critical. If you don't do the conversion or centroiding, the data will be gig level, so you cannot upload or transfer easily. So this is the first step I want to emphasize. So you can select also different format from here. So it's up to you. We support different format. So now I want to show you something on MetaboAnalyst. So the most common one is the 3W MetaboAnalyst.ca. This is our main site. We can click here to start. For the first module is here. This is the module used for raw data processing. We click this. We will get the LCMS Spectral uploading page. You can select a different. You can select your data from here. For example, the most commonly used approach is we need to zip our raw data file into a single zip file. So we have here we have 10. So we zip them in 10 individual files. We can select all of them. This is the file, and we can also select the different select a metadata file. This data file used to describe the group information for our data. So, but here I don't want to go in the data uploading because it's going to be slow, because we have around 80% here. So I want to use our example data directly here. The data have been loaded in our server, so we just need to click submit directly. So as you can see, the data is being uploaded and being loaded by our server. It will take a few seconds. So when you're doing the real demo, I really recommend that everyone use the first example also, because it's a very small data. You use for learning purpose. You can quickly learn how to use a MetaboAnalyst for raw data processing without worrying about the data uploading or something. You don't need to worry. You don't need to wait for hours or several minutes to get the final result. So here, this is the data integrity check page. We can see this is the data file we have uploaded. We can show the centroid have been finished by ourselves. So this is side of the file. We have also provided the group information. If your data is not completely centroid, you can click the convert here button here. But here all data have been centroid, so this is disabled. You can also select different files. If you don't want to include them for the further processing, you just click it. It's going to be excluded. But here I want to include all of them. So in this step, we can click next to go into the parameter setting page. Here we provide user two options. They can use they can select a different parameters setting have already been embedded in our server, and they can use a different mode. The first one is the default. They can use the default parameter, or they can manually change whatever parameter they want to change. If they are an advanced user, they really understand the meaning of different parameters, or they have some experience with these parameters. Or if we are just a clinician or something, we don't have too much experience, we use the auto-optimized. MetaboAnalyst will optimize the parameters for yourself. It's going to be the optimal one, and you can view the results. You can view the result later to compare them. So here I want to show you the auto-optimized workflow. I can just quickly submit. So the data have been submitted. It's being processed. It will take around 2 minutes to finish. So after the so let's wait a little bit. So I want to introduce a little bit on this page. So this is the job status page. User could create the different job URLs when you upload your real data. Here we don't allow them because this example data. It's not meaningful if they create a URL for their example data. So they can view the different purpose, different steps when we are processing the data, and they can see that job is running and have some priorities. They can also save the parameters here. For example, if you finish this data set this time, you want to save the parameters that you can process the data later by using the same parameters. And you can download the status text. It's going to be a text file. You can save all of them for further reference. Here we can see the data is running here. It will take a few minutes. So you can see we are running the DOI step. It's optimizing the parameters. So we can see the Gaussian shapes every time we finish the data optimization. Yeah. Sure.
So in the data can you are uploading, it already appears the names for each group, for example, control, sample,
Yeah.
etc. In each stage, do you enter into that information?
When you upload the page, you need to provide a metadata file. It's a text file, including the information. Yeah.
So the question is, can you repeat the question?
The question?
The question I did was, because when you was uploading the data, there was a column with the names of this, for example, sample, quality control, blanks. So I was asking him in which stage he was entering with that information for each sample.
Yeah, so MetaboAnalyst is designed for statistical analysis. So it's not just for spectral processing. Even you upload data, we ask you provide some replicate. In this case, we have like 4 control, 4 disease, and 2 or 3 QCs. So this one actually is your sample plus the sample group information. So you're going to provide text file, have your sample names, and its interpretation, basically the group label. So you can have QC. If it's QC, you must use QC, which is special term we don't recognize. We're using QC, broad QC for doing the optimization, because QC we found have the strongest signal because it's broad and have more peaks. So we actually use the QC to do the parameter optimization. So if you don't have QC, we're going to use the guess which sample gives them probably largest size. We use that one, have the most signals to optimization. We don't use if you upload one of the spectral, we don't use all of them to do optimization. We just pick the typical one, 5, 3, try to do optimization. So this is that purpose. If you provided the information after later, you can use the functional analysis because you need to compare the group.
So the
yeah, sorry, next time. Yeah.
so the data processing has been finished. So we can see the series become finished. There's going to be a sentence telling you everything has been finished successfully. So we just need to click the proceed. It's going to show the result here. So this is the 3D plot showing you everything. You can use your mouse to rotate or show the different things. This is the loading plot and score plot by showing according to the features. You can double click the button here. So for example, this one. Yeah, I need to use my mouse because of my computer issue. So
You can switch that one. So you can loading switch.
yeah. So we can use the loading plots, and we can see the different features also here. So this step going to be slow. So you can just try your best by exploring this step. So, or we can view the results by clicking this. For example, from the table page, we can view the TIC, total ion chromatography, by clicking the view button, or we can explore the EICs by clicking the this view page. We can also, for example, interactively click the node directly. It's going to plot the EIC for this feature directly from the different samples. As you can see, this is the one from this one. The blue one is from here. They can interactively generate for your manuscript or whatever. So here also the potential chemical IDs can also be provided. Here you can see the potential chemical identities from here. After that, we can go back to the top to see the other results. This is the intensity stats for the different samples. This is also the retention time correction results, showing you the non-linear model works. And this is the TIC plot. This is the base peak ions plot. This is the aligned base peak plot. As you can see, compared to the unaligned results, the aligned one have aligned the variation of different samples. So you can download the result from this page directly, or you can go to the download page. Here is everything you have generated. You can click them to download to your local, or you can start a new journey from here, go to the different modules. So this is something for the further section. So this is a quick demo. I would like to go back my slides. So I finished the demo. So for our coming version, MetaboAnalyst 6.0, we are going to including another dimension for the raw spectral data, MS2. We will provide more functions, so we can see it next year. So I will give you some learning questions in the coming maybe 12 or 20 minutes for learning. So here is the caution when you're doing the practice. You can use the first example directly. The second one is the slow. It will take you a lot of time and avoid downloading and uploading the raw data because we have 100% here, and our server will provide a job scheduling mechanism. If you upload a lot of jobs, you will be scheduled maybe too much time later. And the default MetaboAnalyst includes 3 nodes. 3W is the main site. You can use it directly. For the other 2, you can use it also. Just avoid the too much connections to the main server here because we have a limited bandwidth. So you can doing whatever. I will work around. You can try to use the raw spectral module right now. If you have any question, just hands up. I will go your side. Thank you. Is this working? Sure.
So yeah, we only have 10 minutes because our next statistics, we need to be on time. So feel free to explore using our example data, and actually you can do almost anything you want to follow our tutorial stuff. We will try to help, but the scheduled lecture will be follow what we said, raw spectra, stats, function, and stats too. So feel free to explore. We know we don't have time for too much, but yeah, this 10 minutes belong to you. Yeah, thank you.
Okay, we're going to move on to the next section on statistical analysis for simple experimental designs. Can everyone hear me? Am I speaking into this at the right level? Yeah, okay. So we're uploading these slides right now. They should be available in a few seconds if you want to follow along. So we showed this earlier, the 4 main steps of a omics data analysis. Chung just covered the raw data processing, or the raw data pre-processing. I'll now talk about the data processing and normalization, the statistical analysis and visualization, and then afterwards, Yao will follow with the functional interpretation. So I think that this lecture in the schedule has 20 minutes, but we have a lot of ground to cover, so it might take a bit longer. Later on, it's a little bit shorter, so we'll have more time for hands-on work after that. So the first main step here is data processing. So here we're just trying to prepare our data for the main analysis. Some general steps. First, we look at the sample level, and we're just trying to do quality check, make sure everything went well in our data collection and our raw data pre-processing. We have to deal with missing values. We have to filter our data, so those steps are both done at the feature level. And then we'll do normalization, which considers both at the same time. So quality checking is the first and most critical step before we start our analysis. If we have some samples where something really went wrong, they can really skew our entire analysis. Having high quality data mostly depends on good experimental design and good laboratory practice. There's limited things that we can do once we're actually analyzing the data, but there are 2, and so we'll go over those now. It's related to outliers and batch effects. So one of the first steps when we're analyzing the data is to try and visualize the whole data set and see if there are any major outliers. If there really are truly technical outliers, if we don't remove them, they will mess up all of our statistical analysis. But this is actually an extremely hard question because it's always difficult to say whether an outlier is truly an outlier and due to technical reasons, or if this is just some actual real biological signal. This sample is just interesting. A good rule of thumb is if you see an extremely clear pattern like this, where all of your samples are together and then there's only one single one that's very far apart, and here you can see in the heat map too, that's that one that's a solid red line. I would remove this one. But even if you just had one more sample that's in between that one and that big group, then all of a sudden it's a toss-up, and it's up to you as the experimenter to go back and look at your data and talk to the people who collected the data and try and figure out what was happening. But this is a very difficult question. One point here is that it's mainly related to sample outliers. So we're looking for whole samples that are systematically different than everything else. We don't really care about feature outliers. So if we're looking at one individual feature, we expect there to be a lot where there's a specific metabolite that has some outlier values. This is normal, and actually these are often the ones that we might be the most interested in. The second thing that we care about when we're doing this overall quality checking is assessing for batch effects. So this is just exactly what it sounds like. If we do the actual measurements in different batches, we often see overall systematic differences between batches. These are due for technical, not biological reasons. I have some examples listed there, but often we don't know what's causing them. And if we don't account for these in our statistical analysis, we'll greatly decrease our power, and depending on the experimental design, we could have incorrect results. So this is just the most important thing ever. If you are collecting data, you should never, ever, ever send your samples to the people who are actually running the machines in groups, because if they accidentally do this and run one group as a control, for example, and then the second batch are all drug A and the third batch are all drug B, in the statistical analysis, we have no way of saying whether the differences are because of our experimental design or because of batch effect. And I've seen this personally happen where somebody didn't know this, they sent all their samples in 2 batches, the technicians just ran them, and then you get this. It's basically you can do nothing. So the best design is to randomly allocate your different samples to the different batches with a mix. When we do this, we can apply some statistical corrections and we can consider batch properly in the statistical analysis. So yeah, just to reiterate, this is very bad. Don't do this. This is good. Do this. There are many different batch effect correction methods. I'm not going to go over them in detail, but MetaboAnalyst does have a batch correction module that you can use to use a bunch of them. It's separate from the main workflow. There's kind of 2 different classes of strategies. One is to actually adjust the data itself, so to actually perform an adjustment on your omics, your metabolomics matrix. This can be helpful if you want to use the data for a lot of visualizations like PCA or heat map, because if you don't do this, you might just see the big batches in your figures and you won't see the patterns related to your data. However, if you're doing statistical analysis, you're trying to find features, there's been some work that suggests that it's actually better to just include batch as a covariate within your statistical analysis. There's some work that suggests that works better than trying to adjust your data and then analyze it. So we will cover how to do that later in section 4. All right, the next topic are missing values. So this is really common in metabolomics data. You saw just on the example that Chung showed, there were some NA values there. This is a problem because a lot of the different statistical methods that we use just won't work if we have missing values in them. So we have to deal with them. There's 2 kind of options. Some people throw out any feature that has a single missing value. This loses a lot of information, and I think it introduces more bias. I think it does more harm than good. So in general, we want to guess reasonable values for all of those missing values. In order to do this, we kind of have to understand why the data are missing, because this helps us choose an appropriate strategy. So if you took kind of an undergrad statistics class, this is probably something you covered. There's 3 different types of missing values, and there are different algorithms developed to adjust for each one or to impute for each one. So the first one is simple, missing completely at random. It's exactly like it sounds. It's just like the machine randomly fails, and there's no pattern to why your data are missing. That's not usually the case for metabolomics data. The second is missing at random. So this is there's some randomness here, but it depends on sample characteristics. So a usual example for this is given for like clinical data that's collected at checkups. The younger you are, the more likely you are to have missing values because you go to the doctor for fewer checkups compared to when you're older. And so in this case, the idea is if you're able to get all samples that are the same, so all people that are the same age, for example, then the data would be missing completely at random, but it depends on that. So again, this is not usually something we see with metabolomics data, but there are a whole set of methods that deal with this. So metabolomics data is mostly missing not at random because it's dependent on the actual feature that we're measuring itself. It's just a limit of detection issue usually. It's because the true value is maybe below our detection limit, and so that's why it's missing. And so we're usually going to try and replace that with values that are near the limit of detection when we're trying to do the missing value imputation. And so those are most of the methods that are in that screenshot right there. Okay, the next step is feature filtering. So especially if we're doing an untargeted analysis and we have like 10,000 peaks or something like that, not all of the features are informative. And there are a lot of redundancies in omics data because we're dealing with a biological system. A lot of features and metabolites are correlated with each other. if we filter out low quality or non-informative features before the statistical analysis, we can greatly increase our power. So there have been papers that have shown that you can throw away up to 50% of your features without decreasing the number of significant metabolites that you detect, or like without throwing away much true signal, but you're increasing your statistical power. And I'll explain why when I talk about hypothesis testing in a little bit. So we would feature filters for 3 different reasons. One would be low quality. So this is where if we just have too many missing values, we would throw away that feature. If there's just way too many, we can't accurately impute values for that metabolite. Another is low abundance. So again, these are more likely to be close to the limit of detection and maybe not so reliable. And then ones that have low variance. So variables that are at a near constant value. These are highly unlikely to be related to our experimental metadata, and as long as we don't consider our class labels, like our treatment and control labels, and so we don't filter features based on some statistic that uses those, like fold change or p-value, we can do this without like biasing our statistical inference. So if we're using these totally unbiased methods, just calculating the variance across all samples and then throwing it out based on that, that's okay. So the next step is normalization. This one is hard, but also easy. So there are just some basic things here, like most statistical methods that we use work best when the variables are normally distributed, but most physiological measurements are right skew, like in the top. And so it usually makes sense to perform some sort of transformation, like a log transform, to get things normally distributed. We also sometimes, like, especially when we're measuring samples, we might have different starting volumes or some different thing about the measurement, and so our whole distribution might be shifted a bit. And in that case, some sort of centering method is helpful to try and align all our distributions together. There are custom things that you can do as well if you have some known parameters like dilutions or tissue volumes. There are so many different methods for normalization. The kind of general approach is to use the simplest method first, see if it generally looks okay, and then if you need to do something more complicated afterwards, you can do it then. MetaboAnalyst has a lot of different methods. I'm not going to go over them all in detail. They fall into these 3 different categories: centering, scaling, and transformation. This paper cited down there goes through all of them in detail if you really want to learn about each one. But I'll just show you what I do like every time when I do the demo. All right, so next we'll talk about the actual statistical analysis. So at this point, our data are kind of cleaned and fully ready to analyze. There should be no outliers, no missing values. We filtered out our low quality and uninformative values and normalized and transformed our data. And now we want to identify features that are interesting in our research context. So this section is for the simple experimental design. So one example would be if we have 50 samples, 25 are control, and 25 are treated with some drug, and we just want to find metabolites with different abundances between these 2 groups. So most of the approaches that we'll use, and you should basically always do this analysis first, is univariate analysis. This is where we test each feature independently. We just consider each metabolite one at a time, and we perform some simple method, like a t-test if there's 2 groups, or an ANOVA if there's more than 2 groups. In section 4, later on, we will talk about linear modeling, where we can consider multiple factors, covariates and time series and things like that. Our results from our simple analysis are just going to be a table like this. So each row is a metabolite, and then you can see the t statistic, the p-value, the negative log 10 of the p-value, which just a higher number is better, and then the FDR, or the adjusted p-value. And I'll explain that as well later. And just like the one that Chung showed earlier, all of those have a little visualization button so you can see the individual plots. All right, so one problem with metabolomics data and all omics data is that we are performing these t-tests or these ANOVAs potentially thousands, maybe even 10,000s of times. So our p-value is just this probability that the observed difference between groups is just due to pure chance. There's actually no effect, And we reject the null hypothesis that the p-value is less than our significance level. So if we're doing like 10,000 of these tests, we actually expect to see 500 significant metabolites just from random chance because of how we're setting our p-value. If we think about that 0.05, right, that's the probability of seeing a false positive. If we're looking at just one metabolite, the probability of seeing at least one false positive is just 5%. But here you can see we've looked at 20, and the chances of us seeing a false positive are already above 60%. And so this is why it's super important for you to adjust your p-values and to consider these when you're reporting your results. The simplest way to adjust the p-value is using the family-wise error rate. This is where we just take our significance level, that 0.05, and we divide it by the number of metabolites that we're looking at. This ensures that the probability of seeing at least one false positive stays at 0.05. But this is actually extremely strict, and it's also a little bit wrong if we're thinking about correlated features, but that's another discussion. The more common one is this FDR, which is the false discovery rate. It's a little bit less strict. The false discovery rate is just saying the ratio or the percentage of all of our significant metabolites, what percentage of those are false positives. And so we say, okay, we're actually okay if like 5% of our metabolites are false positives. That seems like an acceptable number. And so if we set our FDR at 0.05, that should be the interpretation. That if you have 1,000 significant metabolites, 5% of those we expect to be false positives after we do the correction. And this kind of gets a nice balance between not throwing out too many true positives without just completely looking at noise. This will be called the FDR or the adjusted p-value or the q-value, depending where you see it, but those are all the same thing. All right, another very common thing that we do with this data is to look at a principal component analysis plot. You've probably all seen a million PCA plots. I know for myself, I probably had to have it explained to me like 10 or 15 times before I really understood it, and I feel like each time I got something else. So I'm going to give you my explanation. With principal component analysis, the goal is to project this high-dimensional data. So every single metabolite is a dimension. So if we have 5,000, we have 5,000 dimensions in our data. And we're trying to project this into lower dimensions that capture most of the variance in our data. So just for a visual, what that looks like. If we consider just 3D space, so we only have 3 dimensions, X, Y, and Z, and we have all of these sample points suspended in there. What PCA is doing is trying to find a new axis that captures the most spread in the data or the most variability. So maybe it's this. And you can see I have the original X values there. Maybe the principal component 1 scores of this new axis are here. So we're trying to find the one that spreads the data out the most. This is integrating information from all 3 original variables, right? So a PCA component score, that's that point floating in that, or the integrated score that we're calculating, is just the loadings times the data. So when we're trying to find that axis, what we're trying to do is we're trying to find a set of coefficients. So in the top right there, it's that A, B, and C. That, when multiplied by our original data, give us these scores that maximally spread out. So that's important to know because we can interpret all of these results. This is showing a PCA plot for samples that are from 4 different groups, the green, red, blue, and brown. The one on the right is showing the actual loadings of the individual features. So you can think about it, we're just showing the kind of extreme ones here. The ones in the top left, for example, those loadings are sort of responsible for pushing the samples in that direction on the score plot. So if we want to know why those green samples are in the top left corner, they likely have really high magnitude values for those loadings in the top left corner. So that's if we see a really interesting pattern in our PCA plot, we can examine the loading plot to try and understand why those samples are in the location that they are. You should get really similar results compared to your t-test. So like if you see a PCA plot and you have all your control and you're treated here, and then you see the loadings that are pushing in those directions, they should be really similar to the ones that are in your t-test results because they're both linear methods. But the nice thing about this is that it groups them together. So you can see like all the group of the features in the top left corner, those are all like have probably correlated abundance levels, and that's why they're pushing in the same direction. So for summary, principal component analysis integrates information from many variables into few variables. It's widely used for a data overview, so it's a really good way of visually detecting outliers and to find relationships between variables, which ones are more similar to each other. It is a linear method for dimension reduction, and there are also nonlinear methods, especially when you start dealing with bigger and more complicated data sets. I'm not going to go over them, but those are testing a UMAP if you're interested in them. All right, so another method in MetaboAnalyst that's available is PLSDA, which stands for partial least squares discriminant analysis. It produces plots that look very similar to principal component analysis, but they're calculated in a different way. You can see these are two data sets, so the one with 2 groups and then one with 4. The principal component plot is the first one, and then the PLSDA plot is the second, and you can see we see much greater separation between our groups. That's because in PCA, we found those loadings, so those coefficients, that computed scores that just maximized variance within the data. So we didn't consider any metadata or class labels when we did that analysis. We just looked for ones that spread out our samples the most. In PLSDA, on the other hand, we find loadings that compute scores that instead of maximizing just unbiased variance, we're maximizing variance between these group labels. We're basically trying to find the loadings that maximally separate our samples in that plot that I showed you. And that's really important to know because it means that PLSDA is always going to produce some separation or more separation than a principal component analysis. And we need to do some extra work to see if that separation is robust, statistically significant, or if it's just completely noise. So PLSDA is susceptible to overfitting, especially when we have small sample sizes. If we have only 5 samples in each group, and we have 2 groups, and we're trying to use PLSDA, I can guarantee you that we'll find beautiful separation, but the chance that those loading scores actually mean anything is really low. So we can use 2 approaches to see if it's like a real separation. One is cross-validation, and I'll explain that. And then the second is permutation tests, which is just another method that's available in MetaboAnalyst. So in cross-validation, the idea is to split our data and our samples into different groups. It's most common to do 5-fold or 10-fold, but I have 3 because it's easier to make the plot. For example, with 3-fold cross-validation, we would take all of our samples, split them into 3 different groups, and then we would rerun PLSDA 3 times. Each time, we would leave out a different group of samples. And so we would use 2/3 of the data to fit our, to find those loadings. And then we would use the third one to assess how good of a job, like the separation is reproduced in our left out test set. We do that 3 times, and this allows us to calculate a bunch of metrics that show kind of how robust the loadings were in our left out. There are 3 different ones: accuracy, R-squared, which you should be familiar with, and then Q-squared, which is sort of a transformation of some accuracy statistics to make them resemble R-squared. This is an example of one that worked pretty well. So you want your Q-squared, that light blue, to be pretty similar to the R-squared, so in the pink. As long as that's happening, you can be sure that the separation that you observed in your data is probably robust. It's quite common to see that Q-squared as like very close to 0 or even negative values. If you see that, it means that you should not put that picture on your poster or in your manuscript because it is not real. All right, so now I'm going to go through an analysis and just kind of show and talk through the steps because there are a bunch of things that I think make more sense if you kind of see the interface. People can feel free to follow along at the same time if they want. And I think we do have a mic here, so if people have questions, I think, Jeff, are you going to run around? Okay. So just raise your hand if you have a question while I'm going through the interface. Right, so everything that I covered is in this module right here. This is probably the most commonly used one in MetaboAnalyst. There are a lot of different data input formats, but we're just going to go down, and I was going to use this example data number 3. All right, so this data were from 50 urine samples from a control group and a group with severe kidney disease. So let me see. The data looks like this. So in this one, the samples are in the rows. So each sample here is a row. Then we have the class information. So we have patient, those are the patients with kidney disease, and then the control, those would be healthy patients. And then we have each metabolite in a column here. You can also format it the other way. You can put your samples in columns and your metabolites in rows. You just have to tell MetaboAnalyst what you did. All right. So I'm just going to submit this. There are no missing values in our data set, so this is grayed out. But if there are, you can click this and it'll show that interface that I showed earlier where there's like 10 or so different methods for imputing the missing values. All right, so this is the first step where we can be filtering our data. The most common way to do this is you can filter just based on QC samples if you've indicated some QC samples in your data. What I usually do is the interquartile range. So this would do something like calculate the variance of every single metabolite, and then we're going to filter out right now the 5% that had the lowest variance. So you can increase this. Oh, you can increase it to 100%. Okay, you can increase it if you want to filter out more. And if you're doing untargeted data, this is where you would be increasing it. Like I would go up to 30 or 40% if I had untargeted data, but if I have targeted with a couple hundred, then I would probably not filter very many at all. It all goes back to that multiple hypothesis correction. I talked about the adjusted p-values. If we have 10,000 features, we're going to be adjusting a lot. And if we're keeping things in there that we know are garbage, then we're just performing this more intense correction for no reason. So that's why you would want to be filtering out here, because it means later on you're going to be adjusting your p-values less. That's why your statistical power increases. You can also, instead of the variance, you can be filtering based on any of these other statistics. All right, so this is the normalization page. It's kind of scary because there are a lot of methods. They're organized into 3 groups. So one is that sample normalization. So this is what I talked about before, where it's where maybe we have differences between our samples related to their isolation or preparation. And so we perform sample level corrections that adjust the entire distribution of metabolites so that they're more comparable. The second one are these data transformations. So the most common one is log transformation. And then data scaling. And I'll show you what this all does. So what I usually do is I first click normalize with just none, because then I can look at it and I can see what the data looks like before I perform any corrections. So this is the feature view showing each different metabolite. We can also look at sample view. So this is looking at the distributions. So whenever we see this kind of pattern where we see like a really, like all of them are like concentrated on the left side and then we have some way far on the right side, that means it's usually a good candidate for log transformation, which is almost always, unless your data was already log transformed. So that's why we check, because if we log transform it twice, that is kind of nonsense. So I'll just log transform it and then we can look at it again. Now you can see it's different. There's more of a spread. It's more normally distributed. This might even be a little bit over transforming. Now we almost have a little bit left skew, but this is not too bad, so I'm going to leave it. You can see these, each sample level distribution, they're slightly different across, but this is pretty good. I would be comfortable moving forward with this in the analysis. If you saw box plots that were all like kind of making a zigzag, they're really all over the place, then it's really important that you perform one of these sample normalizations. So again, the most common ones are normalization by sum or normalization by median. I usually use this. You can do it specifically by some feature in your data, like a reference feature or a reference sample, but that's going to be really individual and depending on your specific data set. So I'll just, if I did this, it looks like this. Again, the sample level. You can see now we normalized by median. This black line in each box plot is the median. And so you can see now they're exactly the same. We've basically just forced them all to be exactly the same. Okay, and then scaling, this is where we, again, you can mean center, which is where you would line up all of you perform this on features. I can just show you again, if we look at the feature view, those should be all over the place. And then if we mean center, oh, yeah, now these are much more aligned. There are specific methods where this can be helpful to do scaling prior. So if you're doing a clustering analysis, it can be helpful, but usually I don't if I'm just doing a statistical analysis. So this is kind of the most common setup that should work for most data sets: normalization by median, log transformation, and no scaling. Okay. I also, yeah, can you tell me if it's time? Okay. Now I'm going to just proceed. I lost my mouse. Okay. So next, this is the overview of all the different statistical methods. I'm not going to go over them in detail, but at the top here, we have this univariate analysis that we talked about. The two main ones are the t-test, ANOVA, and volcano plot. So t-test is going to be available if we have only 2 groups in our data. ANOVA is going to be available if we have more than 2 groups. I personally like the volcano plot. So this is going to plot our data. Along the x-axis is the log2 fold change. And then on the y-axis is the negative log 10 of the p-value. So if those values are higher, that means that p-value is more significant. And then we've applied some thresholds. So we're looking at if the fold change is 2, we're saying it has to be greater than 2 for it to be significant. And we're looking at a raw p-value less than 0.1 here. So I'm going to change this and put the fold change to 1. That basically means we don't care about fold change, any effect size. We're not filtering by fold change. And then I'm going to put this to the false discovery rate. And then click submit. All right, so now you can see we have more significant features because we're not applying that fold change threshold. And then these are the ones that have an FDR corrected p-value of less than 0.1. And I think we should be able to, yeah, if you click a point, you can see the features for that specific one. And then if you look at this table, those are those statistics that you can download. So that will give you each of your metabolites, the fold change, log2 fold change, and then the p-value. I just wanted to go to PLSDA because you can see, right? Here we see some scores plots. The separation looks pretty good. If we go to this cross-validation and we update it, you can see our Q2 is negative or 0. So this would be a great candidate to not put on a poster. And I just wanted to show this because we see a lot of this, like when we walk around the poster hall, we see a lot of PLSDA plots that have really low sample size. Okay, that's all that I have for the demo. If you go back to the home page and you go to this statistical analysis 1 factor, there are many different example data sets. So here's where it's your opportunity to go there, select different ones, try and go through the analyses, and then if you have any questions, we'll move around and answer. So especially if you've used MetaboAnalyst before and you have some things you're not sure about, now's the time to ask and we'll come help. Yeah. Hey Jeff, he has a question. You wanna?
Wow, I got the microphone. Well, that's really good talk. since you mentioned batch effect, I'm just wondering, can you guys do mixed effect model?
Yeah, we'll talk about it in section 4.
Thank you.
Okay, agora sim. What we should to do with the zeros in the table? Typically, we can have variables with a lot of zeros in the table. So these zeros are regular data, or we can treat these zeros as a missing data.
Oh, so the data is your data, and in a single cell or microbiome with count data, we do see a lot of the zeros, which basically it's called sparse data. We don't see them, but we do expect it if we able to sequence in deep. So there's definitely a method to deal with many zeros. But in metabolomics, and I don't know what type of data you get, but for human like blood and urine, and we detecting, especially targeted or even this stuff, we do see zero, but not that many. If we, in MetaboAnalyst assumptions, if it's zero, we think it's something below detection limit, and we try to treat them as missing value. This is default reasoning. But if indeed in certain samples we need to treat them differently, definitely there's other thoughts, but this is default thinking. Okay.
Sorry, can I just follow up on that question? I guess I'm just wondering, what is your opinion regarding MCMC-based kind of like multiple imputation method?
Markov model?
Yeah. Since this kind of multiple imputation has been used in many other kind of omics, for example, genetics, like things in the underlying genotype markers, like those kind of imputations. So I'm just wondering, what's your opinion regarding on this kind of thing?
On what application? MCMC on?
But for example, for like large study, population study, for the genome marker.
For genome, for missing value imputation?
Yes.
You want to say something? Yeah, come now.
I know I've used some of those methods before, but they take a really long time to run, at least. So I use them in R when I'm analyzing my data set sometimes, but we can't put it on MetaboAnalyst because it would probably crash the server if a lot of people did it at the same time. So I think like the more simple methods, they work pretty good too if you're using some of the ones that have some more randomness in them. But if you wanted to use those ones, like you probably have to do it locally just because some things are just not really work well to do on our server.
The computing is one reason. There's a strong belief in metabolomics. A lot of the missing is due to a detection limit. So we just use the simple but making sense. MCMC in genomics or other part, we do see it. and we do think that's reasonable in metabolomics. I need to be convinced. That's one thing is about computing. The other one is rather the randomness or that process is meaningful compared to the simple ones. Because if we have simple ones, very relatively easy to interpret. That's so good, and a lot of time it's working well. So in summary, if you believe that's useful, publish a paper, show the convincing evidence. Because there's a lot of things I'm doing a lot of different omics. I see different omics data actually quite suitable for different approaches. Some approach, it's just a table, you can use it, but it's not working well in metabolomics versus the RNA-seq data. So this part is, we don't know why, but really, the cross-border application of certain method is quite something I would like to do, but waiting to see the evidence. There's something like 20 years of using PCA in metabolomics or chemoinformatics. It's just working well, and I see it working well. But when we apply to microarray transcriptomics, it's not as so effective. It's something it's fundamentally make some distribution of data looks different. And sparse data is one thing. Microbiome, we account a lot of single-cell account, but metabolomics, I don't see a lot. So, but I hope in the coming years, when we're doing a lot of multi-omics data, we're probably going to find some themes and recommendations. And at the moment is, we have a lot of tools in the box, but it's selection of them. I don't want to just put it there unless we clearly know generally it's useful, right? And if you are interested in certain method in this module or other module you want us to demo, we can also do it. It's just depending. If you enjoy what you're exploring, and just do it, or ask other questions, or you want us to demo, we can also do it. So this is still the hands-on time. So it's your time. You can make a request.
Okay, so fantastic talk. I really like the emphasis on the cross-validation for PLSDA. I wanted to ask you about some of the other PLS, the scores plots that are generated here, the sparse PLSDA and ortho. If you could talk about potential overfitting with those, and if you have cross-validation methods for those as well.
Yeah. Jeff, can you answer that one? I didn't implement those.
So let me your question is about whether sparse PLSDA so far it does not contain the cross-validation. That's you.
Yeah, there's questions about overfitting because I know PLSDA tends to do that, and I know like ortho is also notorious for that. So how do we?
Your question is compare sparse PLSDA versus PLSDA.
Comparisons between them, and then is there a way to cross-validate those to know if the results we get are overfitted or not?
That's a good question. I think, how to say, I read a paper by Tobias Kind from UC Davis, and I recall the conclusion is that disregarding how you're doing it, PLSDA probably fundamentally is the same. There's some visualization difference, and but overall, if whatever we have done in terms of classification, PLSDA result is very more or less stays. So this is a PLSDA versus the old PLSDA. But the sparse PLSDA, I don't see anybody have done that benchmarking yet, to be honest with you. And it is relatively new method. I don't feel there's sufficiently people using it to show it's doing very well. So this is, do you have some experience in your observation and motivating you ask this question? Because the PLSDA, yeah. we do have the consistent implementation for SPLSDA versus the PLSDA. But you are talking about the performance, right? Whether it's SPLS perform better.
Sometimes I see a little better separation with sparse. And my question is that real?
Yeah, do a more study and publish a paper. I will be happy to. So be honest with you, MetaboAnalyst is really based on the community report. If some method is really doing well and people get a good result and we gain confidence, we just make it available. And sometimes we do have time ourselves to do benchmarking, but it's really, really depending on how much extra time we have. If this one is really, really common needs, we may spend more time. But last time about chance stuff, we realized the XMS default use is really a bottleneck. So we spend a year to make it automated. And there's certain things need more benchmarking. We're willing to do it, but this one is new to me. And send me the paper. If you get a good paper, you really believe that method need to be used by the community more widely, and we are willing to consider, okay?
I know for the orthogonal PLSDA, there is a permutation tab. So that is doing a whole bunch of permutations to test for if there's like no effect. So that can be helpful. I did just look at the sparse PLSDA, and they do have a cross-validation tab, but it gives different statistics, so the interpretation would be a little bit different. Yeah, it's showing the classification error rate like based on the model with cross-validation, so that can be helpful, but we should put better guidance on there for how to interpret this to know if your data is garbage or not. Or not your data is not garbage, if the model is not good for that data.
Thanks for the presentation. Just a general question about, I work with the food animal species as a veterinarian. So those are the species, like the different strains, they are very homogeneous. And how do you really get the sample size? You just based on cross-validation, permutation, and you go with the increased sample number, or how do you really get the cutoff? Yeah, this is exactly the N number should be sampled with a 20 or 30, that kind of thing. Compared to a human, for instance, like they are very heterogeneous.
Like, so you're asking what sample size you want and things. There is a power analysis module in MetaboAnalyst. Maybe Jeff can talk about it. I don't know so much about it, but it's for answering those kind of questions.
Yeah, this is very hard to answer questions. But really depending your effect size. In general, metabolomics, I see if you want to do like a clinical study and with reasonable effect size, and we see like 20, at least 20, 13, 14, I think is a common one. But if you study like model species like E. coli and C. elegans well controlled, you just have 5 and 5, you can clearly see the result. So really depending on how strong the effect size, how homogeneous it is. And the power analysis is really designed for more human and population study. And you can just upload a pilot data and see the effect size. If you really want to see the power like 18% or 0.8, and you will get what's the number of samples. If you're balanced per group, we'll let you know. So this is something is you need to see a similar study from your publication, what's the number. But if for the MetaboAnalyst, we just discussed about the PLSDA cross-validation, and I think minimal, in my opinion, is 12, okay? 12 per group. Other than that, you don't even have enough to permutate or doing cross-validation. So all the stats you want to do, it became just not applicable. So if you really have a small number of sample, the only way you can do is fold change and t-test, almost like that. And you want to do some heavy lifting like deep learning, there's no way you can do that. So deep learning require almost hundreds of samples. Yeah.
Yeah, these are the work that I do, and then we use the groups for the real clinical studies, depending on the clinical science or mortality, whether it's infection versus control. In the real animal trials, we use 20 as the group size. Actually, we did use for Metabolomics 30. And then we have the clear separation with the PCA, PLSDA. I'm just wondering, because there's no really a guidelines or standards, and that's why I asked the question.
Okay.
So the real animal studies, whether it's the real clear survival, bacterial load, or even metagenomics, 20 is fine, but we increase another 50%, so that is 30. So I'm just wondering, is that enough for the accuracy?
Yeah, like it completely depends on your effect, what you're trying to measure. Like if you're trying to measure something really subtle, like some very small change in physiology, then you're going to need a huge number of samples. If you're trying to look at like differences between tissues, then you only need like 2 samples. Yeah, I mean, it depends what it's a biomarker for, and also how precise your measurement is, and like how the quality of your sample. So the best thing to do is, it's impossible for us to answer without knowing all of those details. So it's best to like try find as many papers as you can that published in a similar technology, a similar like assay, a similar hypothesis, and see what sample size they used, and if they get like a good power in their results.
Yeah, especially
Yeah.
if you're doing biomarker, I suggest close to 100. Okay, otherwise, I don't have any confidence. Biomarker really need a cross-validation and holdout, or basically, we're talking about at least a double cross-validation, because any method we use cross-validation, we know it's overfit. Even we use permutation cross-validation, it's still not strong enough. I suggest you have to have a training, validation, and holdout to do. Even if you're doing applied, almost have a separate separate location to do another one. So we are talking about hundreds or several hundreds. So biomarkers require more sample. Okay, MetaboAnalyst do allow you to do biomarker analysis, but clinical biomarker is really more stringent, should be. Yeah.
Okay, we'll have a last question before the break.
Shyam from Emory University. Coming back to the PLSD analysis, I'm not clear that if the PLSDA, the Q-score is negative, then does it mean that the treatment groups are not significant, and what shall we do next?
Well, I would always look at your univariate results first. So the results from your t-test and your ANOVA, those sort of things. And if you're getting no results there, and you're not really seeing separation in your PCA, and the PLSDA Q-score is negative, then I'd say you don't have enough power to be looking at what you're looking at. But that's why it's helpful to look at the results from multiple tools at once. And if you consistently don't see a strong effect, then you just need more samples for what you're trying to look at.
So sometimes we have a significant metabolites in the t-test or the Volcano plot, but the PCA or PLSD is nothing, right? So are we focusing on the univariate rather than a multivariate, if not significant models created in the PLSD?
Well, I mean, it's just you're trying to interpret all your results together and try and understand what's happening in your data. So it sounds like in this case, it wouldn't make sense to make any sort of predictive model because you don't have the right experimental design for that. And also I'd go back to your univariate tests, and it's common in omics data to have a small number of significant features, but not to see a huge separation in your PCA plot. Because it could just mean that your bigger sources of variability in your data are related to some technical factors or something like that. So
I just follow up on that. You t-test, in general, you should be able to see something, but PCA is fundamentally global, you're multivariate. You see the difference is normal, okay? You cannot try to compare. I have to see some features in t-test, I expect to see the same thing, separation in the PCA. It's not comparable, okay? That's either way, it's you have or have not. These two are very different things. Also, regarding to your PLSDA, if you have sufficient number of samples and you get negative Q-square, that really means that's not reliable. If you have less number of samples, then you probably can improve power by adding more samples. But if you have a lot of samples, you get negative. That really means not predictive, that features. So, yeah. I guess we are on a break for 15 minutes. Coffee. I think coffee is served. Yeah.